Hi, welcome to Fortney's Fantastic Finds. I'm Nathan.
And I'm Charlie.
And today we will be extracting DNA from food products you might have around your house using everyday household items. Charlie, what is DNA?
It is the building block of all life.
Where can we find DNA?
Inside of cells.
Okay. Now, before we get started, Charlie, don't forget, safety first. Every good scientist knows it's important to wash your hands before you go to the lab. Perfect. Now, here's a tube you can use to extract. We're going to use wheat germ to extract DNA from, but you can also use other food items you might have around your house. Like here we have some spinach that's smushed up in the bottom of a tube. Or you could also use fruits like apples or bananas or strawberries. Anything like that will work for this experiment. First thing we need is about 1 gram of wheat germ in the bottom of the tube, which is about 1/4 of a tablespoon. You want to measure that out? I'll hold your tube. Perfect. Okay. Next thing we want to add is warm water. Now, make sure it's not too hot, because that will break apart the DNA that we're trying to extract. But you want it to be warm. So here I have a cup of water that I microwave for about 1.5 minutes on high. And you can use a thermometer you might have around your kitchen, and make sure that it says about 140 degrees. It can be a little bit above or below, but right around 140 degrees Fahrenheit is perfect. Now we need to add some to our tube. We need about 2 tablespoons worth. 1 and 2, and then I'll do the same for my tube. Okay. Now we need to shake this really vigorously for about 2 minutes, okay?
Mm-hmm.
All right, I'll start my watch. Go. Okay, that's been about 1 minute. Okay. That's good. Yep, perfect.
Why are we using warm water?
That is a great question, Charlie. So we use warm water because that helps soften the membrane of the cell. So that's the outside protective layer of the cell. But we don't want it too hot because that will break apart the DNA. Now we need to add a bit of salt. Okay, you can take the tube off. Or here, I'll dump that in my tube. You can hold your hand out. Now we're going to shake for another 2 minutes. Okay, that's been 2 minutes. Now we're going to add some soap and rock gently back and forth.
Why did we add table salt?
That's another good question, Charlie. We just added regular tap water, but we want to add a little bit of salt because that will make the water that we added more similar to the inside of the cell. So when it breaks apart, the DNA doesn't break apart. It's more stable in the water. Okay, so I'm going to add, I don't know, about wasn't really a drop. Now we're going to rock this gently back and forth for about 5 minutes. Okay, and that's our 5 minutes of shaking back, or rocking back and forth. We don't want to shake it. Now, in the bottom of our tubes, and probably in the bottom of yours also, there's still a lot of big chunks from the wheat germ. So what we want to do now is transfer the nice, clean, clearer solution to a new container that we're going to extract DNA from. We'll do a close-up here of Charlie's tube to show you what we're talking about. Perfect. Now, if we look at Charlie's tube here, at the bottom, there's still some debris from the wheat germ that we started with. But what we want to collect is this nice, clear, I guess kind of yellowy looking solution in the middle, and we'll move that to a new container. Here's a pipette that you can use, and here's a glass tube that you can transfer it to. So we want to move about 5 milliliters, which is about 2 of these eyedroppers full. So you can grab it, yeah, just stick it down and grab. Oop. Yeah, that's okay. Yep, perfect. Now I'm going to use a plastic drinking straw to collect from the bottom of my tube because it's a much bigger bottle. But if you have something at home, like an eyedropper for medicine or something, that would work as well. Okay, my straw solution didn't work very well, so I'm just gently tipping the side of my bottle and getting some more of the nice kind of clearer liquid from the side, and trying to avoid any of the chunks from the wheat germ or whatever type of food you're trying to get DNA from. So Charlie's using a nice glass tube. I'm using a little medicine cup that you might have from like a bottle of cough syrup or something. Some sort of small container that you might have around your house. The taller and skinnier, the better. Now we are going to precipitate the DNA. What happens when you put sugar in water?
It dissolves.
Yep, that's right. So DNA, like sugar, dissolves in water, but DNA will not dissolve in alcohol. So we're going to add some alcohol to the top of our tubes, and that will pull the DNA out of solution so we can see a nice cloudy kind of snot ball looking layer of DNA from our wheat germ. We have 2 different types of alcohol that we can use today. This first one is ethanol, and this is often used as like a cleaning solution. So you might be able to find this at a hardware store. Or this is isopropyl alcohol or rubbing alcohol that you can find at your local pharmacy. So Charlie, which one do you want to use? Okay. So if you're using isopropyl alcohol or rubbing alcohol, you need about the equivalent volume of what you collected. So you can see in the bottom of Charlie's tube, there's maybe, I don't know, about 1 inch of liquid at the bottom. So Charlie here will add about the same amount on top of it. And in my little medicine cup, I have about 5 milliliters at the bottom. But with ethanol, you want to add 2 times as much volume to pull out your DNA. When you pull out the alcohol from here and put it in your test tube, you want to make sure to carefully run it down the side of the container. You don't want to just like squeeze it in there, because you want to keep it so there's a layer of alcohol on top and then your wheat germ solution at the bottom. And yeah, slowly squeeze it out. That's perfect. Okay, let's take a look. Okay, yeah. So you can see in Charlie's tube how there's still the wheat germ solution at the bottom, and then all the alcohol that you just added is sitting on top. Now we'll want to carefully set that in another container, and we'll just let that sit for a couple minutes. And meanwhile, I'll get my tube ready, and then we'll look at our DNA.
Why were we adding dish soap?
That is another good question, Charlie. The outside of a cell is kind of like a grease bubble. So think of like washing dishes. If you have a greasy pan and you put a drop of soap in there, where does all the grease go? Does it kind of break apart and spread out?
Kind of.
Yeah. So that's the same idea with the cell. We're adding soap to break that apart. And inside of the cell, there is something called the nucleus, and that's what holds all of the DNA inside of our wheat germ cells. So that, the outside of that nucleus is made out of the same sort of material that the outside of a cell is. So we add soap to help break apart that membrane so we can get the DNA out of it. Okay. Let's take a look at your tube, Charlie. Do you want to carefully hold that up? Okay. So there's a little bit at the top. We'll let that sit for a couple minutes. Now here's the tube that I prepared, and this one I used the ethanol instead of the rubbing alcohol. This kind of goopy looking layer at the top, that's all the DNA from the wheat germ. And if you're really careful, you might be able to pick up some of it. Let's try that again. Okay, now let's take a look at Charlie's tube. So this was using the rubbing alcohol. Do you see that goopy bit there on the side of the tube? Mm-hmm. Yeah, just very carefully try and poke it a bit and scoop it up. Yep. You can try and like twirl, twirl the toothpick like you're twirling up spaghetti. Yeah, that's good. Yep, and then gently pull it out of the tube. Let's. We didn't twirl it up, but there's a goopy bit on the side of the tube, which is all wheat germ DNA. So this is some wheat germ that I extracted with ethanol. So here at the top is more of the goopy DNA. So we'll just try and slowly, carefully spin it around on the end of our wooden skewer. Thanks for joining us today. Hopefully you had success seeing your DNA and maybe even spooling it up on your toothpick. If you had good success with it, make sure to take some pictures and send those in and share with everyone else at Science Expeditions. Thank you and see you next time.
Bye.